When you picture a cancer cell trying to hide from the immune system, you probably just imagine it throwing on some kind of molecular camouflage.
Right, like just blending in with the healthy cells around it.
Yeah, exactly. But what if the reality is much more active and honestly a lot more sinister than that? Imagine a cancer cell is actually hiding this massive stash of red flags, evidence that could instantly alert your immune system to attack it.
But it has this built-in industrial-grade shredder, right?
Yes. It is actively destroying the evidence before it ever gets seen. And that brings us to the core of this deep dive. What if we could just break that shredder?
It sounds almost like science fiction, honestly. But it completely changes the paradigm of how we think about tumor visibility. I mean, we've spent decades trying to paint a target on cancer. What if instead we just stop the cancer from destroying its own targets?
Okay, let's unpack this. We're diving into a really fascinating 2026 paper from the journal Immunity by Vendramin and colleagues, and they suggest we might finally know how to do exactly that.
Yeah, it's a huge breakthrough.
So we've all heard of immune checkpoint inhibitors, or CPIs, drugs like anti-PD-1. For some patients, they are absolute miracles. They basically take the brakes off the immune system, let it go to work. But there's a pretty huge catch, right?
There is. They really only work well for tumors that have a high tumor mutational burden, or TMB.
Right.
TMB is essentially a measurement of how many mutations exist inside a tumor's DNA. If a tumor has a high TMB, say, something caused by heavy smoking or UV damage, it is generating lots of what we call neoantigens.
And those are the red flags we were talking about.
Exactly. They're new abnormal proteins that act as targets for the immune system. But the problem is only about 10% of human tumors are actually TMB high.
Which leaves this massive, frustrating gap. I mean, what about the other 90%?
Right, the low mutation tumors that basically look completely normal to the immune system.
Exactly. How do we force them out of hiding?
Yeah.
Well, that brings us to our first major concept from the source material, which is this idea of a cellular spell checker.
Yeah, to understand the spell checker, we really have to look closely at how cells build proteins. As we know, DNA is transcribed into messenger RNA, or mRNA.
That's the instruction manual for the cell.
Right. And the ribosome reads this mRNA and translates it into a physical protein. But genetic code isn't perfect. Sometimes there are mutations in the DNA. We're talking about things like a frame shift insertion, where an extra genetic letter gets shoved in where it shouldn't be.
Or like an intron retention, right?
Yes, where a piece of non-coding junk RNA accidentally gets left in the final manual.
And these specific types of mutations cause a major structural problem.
They do. They often create a stop sign too early in the genetic code. In biology, we call this a premature termination codon, or PTC. If the ribosome hits that premature stop sign, it literally just stops building the protein halfway through.
Okay, let me try to visualize this. If I'm picturing the cell's protein building process like a factory, the DNA is the master blueprint locked in the manager's office.
I like this, yeah.
And the mRNA is the photocopied instruction manual sent down to the factory floor. So the PTC is basically a glaring typo in that manual that just says, "Stop building," halfway down the page.
That is a perfect analogy. Now, if the factory workers just followed that typo-ridden manual blindly, they'd produce a weird, truncated mutant machine. But healthy cells don't want mutant machines floating around, so they have a ruthlessly efficient quality control inspector.
The spell checker.
Exactly. This pathway is called nonsense-mediated mRNA decay, or NMD. NMD actively scans the factory floor for these premature stop codons. When it spots one, it immediately degrades. It shreds the flawed mRNA transcript before the mutant protein can ever be built.
And here's where it gets really interesting. Because you'd think a quality control inspector is a good thing. Keep the cell healthy, right?
Usually, yeah.
But cancer cells totally hijack this process. The researchers show that tumors often have highly elevated NMD activity. By running that shredder on overdrive, the cancer destroys these weird instruction manuals. It actively prevents these abnormal proteins from being built and displayed on the cell surface. It's actively hiding its own mutations. I've been using a little mnemonic to remember this. NMD equals no mutant display.
Oh, no mutant display. I like that. It is a brilliant evasion tactic. The cancer is basically using a normal, healthy cellular safety mechanism to continuously wipe its own hard drive of any incriminating evidence.
So if I pause and look at what we've established so far, NMD is a cellular shredder that destroys mutated RNA. And by doing its job too well, it accidentally helps cancer hide its mutant proteins.
Yeah.
Which leads me to a question for you, the listener, to think about. If you were a scientist looking at this, would you try to go in and fix the millions of microscopic mutations, or would you just try to fire the quality control inspector? Because we're about to see what happens when you fire them.
Exactly, but you can't just fire them without knowing who signs their paychecks, right? The researchers needed to find the master switch of this NMD pathway.
Right, they couldn't just guess.
No, so they systematically analyzed massive clinical data sets. They looked at the TCGA pan-cancer database, which holds vast amounts of genomic data across many cancer types, and they paired it with the CPI 1000+ patient cohort.
And that cohort is tracking over 1,000 patients who had actually been treated with checkpoint inhibitors.
Exactly.
I noticed in the paper they used something called an NMD score to evaluate these patients. How exactly do you score a shredder that you can't even see?
It's a remarkably clever piece of math, actually. The NMD score is a ratio. The researchers measured the express PTC-containing transcripts, so meaning the mutated RNA that somehow survived and didn't get shredded.
Right.
And they divided that by all the PTC-inducing mutations present in the actual DNA blueprint.
Oh, I see. So if your DNA has 100 mutations that, should trigger the shredder, but 50 mutant manuals still make it to the factory floor, your NMD score is lower because your shredder is basically failing.
Precisely. A low score means the NMD pathway is weak. And what the clinical data revealed was striking. Patients with lower NMD scores responded significantly better to checkpoint inhibitor therapies.
Wow.
Yeah, and that held true regardless of their overall tumor mutational burden.
So the data proves that a naturally broken shredder is incredibly good for the patient's survival. But how do we break it on purpose for everyone else?
To figure that out, the team ran a rigorous ranking system using CRISPR gene editing screens. They systematically knocked out different genes involved in the pathway and compared that with survival data and expression analyses. And the clear winner, the ultimate target to block, was a specific kinase called SMG1.
SMG1. Okay, why that specific kinase out of everything in the pathway?
What's fascinating here is the precise biological role of SMG1. It is the rate-limiting step for the entire NMD pathway. Its specific job is to initiate the degradation process by phosphorylating a masked regulator protein called UPF1.
Right, and in biology, phosphorylation is basically a chemical on switch. You attach a phosphate group and the protein wakes up.
Exactly. So if SMG1 is blocked, it can't phosphorylate UPF1. And if UPF1 stays asleep, the whole NMD shredder mechanism completely grinds to a halt.
Okay, let's take a breath here and do a quick recap, because we are deep in the weeds and I want to make sure we're keeping this clear for everyone listening.
Good idea.
Here's what we know so far. First, low mutation tumors hide from the immune system because they don't display any red flags. Second, NMD, the cellular shredder, is what destroys mutated RNA that contains those premature stop codons. Third, cancer abuses this NMD system to hide. And finally, the SMG1 kinase is the on switch for that shredder. Turning it off might reveal the tumor.
That's the perfect summary. The researchers proved through massive databases that blocking this SMG1 kinase shuts down the NMD pathway. And clinically, a weak NMD pathway correlates with much better patient survival.
Which leaves us staring at a massive, critical question. We know where the master switch is. But how do we physically prove that flipping it actually makes the cancer visible to the immune system? Because, a computer database is one thing, but biology is messy.
Biology is incredibly messy. To prove visibility, you have to look at the surface of the cell. Specifically, you have to look at a structure called MHC class I, the major histocompatibility complex.
I always think of MHC class I as the cell's display window.
Yeah, that's a good way to look at it.
Every single cell in your body has to constantly prove to the immune system that it's healthy and doing its job. So the cell routinely chops up tiny bits of the proteins it's building inside. These fragments are called peptides, and it physically sticks them out in this display window on its surface.
Right.
And passing immune T cells are like cops on the beat, right? They look in the window. If they see normal self-proteins, they tip their cap and move on. If they see weird mutant viral or cancer proteins, they call in the SWAT team.
That is exactly how it works. So the researchers needed to see if blocking SMG1 actually forced new mutant targets into that display window. They treated melanoma and lung cancer cell lines with specific SMG1 inhibitors. Then, to predict what would happen, they used a highly advanced algorithm called NetMHC-PAN.
Wait, how does a computer algorithm predict what a living cancer cell is going to display on its surface?
It's essentially a complex 3D structural matching game. The algorithm analyzes the physical, chemical shape of these newly stabilized mutant peptides, and it calculates the binding affinity, so how tightly they will physically snap into the groove of the MHC class I display window.
Oh, wow. And what did the algorithm predict?
The results were massive. Blocking SMG1 expanded the predicted neoepitope repertoire by 20-fold.
Wait, 20-fold?
20-fold.
So by just stopping the shredder, they mathematically made a low-mutation tumor look exactly like a high-mutation tumor to the immune system.
Exactly. And the most vital detail here is that they achieved this massive increase in targets without causing any new DNA damage.
That's a huge point.
Right, because traditional treatments like chemotherapy or radiation try to create new targets by blasting the DNA and causing double-strand breaks. That is highly toxic. Here, the DNA is completely untouched. You are simply forcing the cell to reveal the typos that were already there.
But again, an algorithm is just a mathematical prediction. How do they get physical proofs that these specific mutant proteins actually made it to the window?
To get physical proof, they had to turn to a proteomics technique called LC-MS. That's liquid chromatography tandem mass spectrometry.
Okay, let's translate that for a second. How do you physically measure something that small on a cell?
It's a monumental technical challenge, honestly. Basically, they have to chemically watch the peptides off the MHC class I windows of millions of cells. Yeah. They separate this incredibly complex soup of proteins using liquid chromatography, and then they blast them in a mass spectrometer. The machine physically weighs the fragments to figure out their exact amino acid sequence. But even if you weigh them, you still have to match them to a database to know what they are.
Right, like running a fingerprint through an FBI database.
Exactly. But here is the problem. Conventional databases only look for normal canonical human proteins. If you search for a mutant, the database just comes back blank.
Oh, because it doesn't know it exists.
Right. So the researchers used a custom database tool called MS-Fragger, which they intentionally loaded with all the aberrant mutant genetic sequences they suspected were hiding.
And did they find them?
They absolutely found them. They physically detected specific non-canonical peptides, for example, one derived from a frameshift in a gene called DDX20, and proved they were successfully displayed on the MHC class I window, but only after the SMG1 inhibitor was applied.
Okay, if I'm following this, we now have undeniable physical proof. Inhibiting SMG1 successfully forces the cell to build those typoproteins and stick them in the display window. It increases the tumor's targets 20-fold without mutating the DNA further.
Yes.
But here's the next hurdle and a question for you to ponder. We have the target sitting in the window, but will the immune police actually care? Because, having a target isn't enough if the T cells don't recognize it as a threat.
You've hit on the crucial next step. The immune system has to be capable of recognizing and killing that specific target. So to prove this causality, the researchers designed a truly genius laboratory mechanism, a dual reporter system.
How on earth do you prove that in a lab? It sounds impossible to track whether a T cell is attacking because of the shredder being broken or just because it's in a hostile petri dish.
It's brilliant, actually. They engineered a fluorescent protein called EGFP, so it glows green, and they fused it to a very famous, highly immunogenic cancer antigen called NY-ESO1. Then they made two distinct versions of this reporter. Version A had a premature termination code bond built directly into it, making it an immediate target for the NMD shredder. Version B was normal. It completely lacked the PTC.
I see. So they have a mutant version and a normal version side by side. It's the perfect biological control to isolate the variable.
Precisely. They introduced these reporters into tumor cells and then exposed those cells to T cells that were specifically engineered to attack the NY-ESO1 antigen. When they applied the SMG1 inhibitor, the T cells fiercely attacked and killed the cells carrying the PTC-targeted reporter.
But what about the normal one?
The cells with the normal reporter saw no increase in attack whatsoever.
That is the smoking gun right there. That proves the SMG1 drug isn't just acting like some broad stimulant making T cells generally angry or hyperactive.
Yeah.
It is surgically and selectively rescuing the exact targets the NMD shredder was trying to hide.
Exactly.
But, a lab construct is still just a lab construct. What happens when you move to real living human tissue?
Well, that was the exact progression of the study. To test reality, they used ex vivo patient-derived tumor fragments, or PDTFs. They took real, living tumor biopsies straight from patients with colorectal cancer, kept the tissue alive in a dish, treated it with the SMG1 inhibitor, and watched what happened to the native immune cells that were already naturally trapped inside the tumor tissue.
And we really need to highlight the specific type of colorectal cancer they chose for this. The paper emphasizes this was 80% pMMR or MSS colorectal cancer.
Right, that's crucial.
Let's translate that for a second. We're talking about tumors that have mismatch repair proficiency and microsatellite stability. Basically, they lack the massive, chaotic DNA repair errors that usually alert the immune system. In the oncology world, these are notoriously immune cold tumors. They are historically incredibly resistant to immunotherapy.
Yes, they picked the ultimate challenge, and the results were remarkable. When they treated these human tumor fragments with the SMG1 inhibitor, it directly activated the tissue-resident memory T cells. We saw a massive increase in surface markers like PD-1,
indicating the T cells were waking up from their dormant state.
Exactly. We also saw increased secretion of interferon gamma, which is a key chemical weapon T cells use to destroy threats.
They also used single-cell RNA sequencing, didn't they? What did that show?
It showed actual clonal expansion. By looking at the RNA and sequencing the T cell receptors, they proved that the exhausted T cells inside the tumor weren't just waking up, they were rapidly multiplying.
Wow.
And importantly, this fierce reaction was isolated. It did not happen in the healthy, normal tissue adjacent to the tumor.
If we pause and connect this to the bigger picture, it suggests something deeply profound about cancer biology. It means tumors are inherently leaky.
Yeah, that's a great way to phrase it.
Think about it. The T cells in that patient's body have actually seen these NMD antigens before.
Yeah.
At some point, the cancer slipped up. The T cells saw the red flags, tried to fight, eventually got exhausted, and were just sitting dormant inside the tumor, waiting. And the moment the SMG1 inhibitor broke the shredder, the antigens flooded the display window, and those exhausted T cells remembered exactly what to do. They woke up and multiplied.
It's beautiful biology. Let's do a quick structural recap of this massive amount of data before we look at the final crucial test.
Sounds good. We're right around that 10 or 12 minute mark anyway.
Okay, first, the mass spectrometry physically proved that the rescued mutant proteins reached the cell surface. Second, the Genius fluorescent reporter system proved T cells only attack when the NMD pathway is specifically blocked. And third, real human colorectal cancer fragments suddenly woke up their exhausted immune cells when treated with SMG1 inhibitors.
So we have undeniable ex vivo proof that turning off NMD wakes up exhausted T cells in actual human tumors. Which leads to our next logical question, and really the most important one for you, the listener. We have petri dishes and tissue fragments, but can this actually shrink a tumor inside a living, breathing organism?
That brings us to the ultimate test, in vivo synergy. To prove this works in a living system, the researchers used an LLC1 syngeneic lung cancer mouse model.
Let me jump in with a skeptical question here. Why this specific mouse model? There are dozens of cancer models they could have used. Did they just pick one that's, easy to cure?
It's an excellent question, and actually it's the exact opposite. The LLC1 model is infamous for having a heavily immunosuppressive, myeloid-rich tumor microenvironment.
Meaning it's packed with the kinds of white blood cells that actually protect the tumor instead of attacking it.
Precisely. It is highly refractory, meaning it is totally resistant to standard checkpoint inhibitor therapy. It represents the absolute worst-case scenario. It is the ultimate stress test for an immunotherapy drug.
Okay, so they took these worst-case scenario mice and treated them with KVS 00001. That's a special, optimized version of the SMG1 inhibitor formulated to actually survive and work inside a living body, right?
And they tested it alone, and they tested it in combination with a standard anti-PD-1 checkpoint inhibitor.
And what happened?
The results were definitive. The combination therapy profoundly suppressed tumor growth, significantly better than either the SMG1 inhibitor or the anti-PD-1 drug could do alone.
Amazing.
Yeah, they used a technique called flow cytometry to sort and count the cells inside the mouse tumors, and it showed a massive activation of CD8-positive cytotoxic T cells.
And how do we know those T cells were actually fighting? Physically doing damage?
Because they showed huge spikes in activation markers, things like 4-1BB and ICOS, which are basically the go signals for T cells, and most importantly, granzyme B.
Granzyme B.
Right. Granzyme B is the actual toxic payload. It's the physical protein that T cells inject into cancer cells to trigger them to explode and die. The SMG1 inhibitor essentially caused the T cells to arm themselves to the teeth.
So if I'm looking at this whole picture, by Combining the SMG1 inhibitor with the standard PD-1 drug, you're essentially breaking the tumor's camouflage while simultaneously handing a megaphone to the immune system. They work together, the shredder is broken, the flags are up, and the immune system attacks a tumor that was previously completely resistant.
Exactly.
This brings us to the core question of our deep dive. So what does this all mean for you, the listener, and the future of medicine?
It represents a fundamental paradigm shift in oncology. We used to think we were entirely at the mercy of whatever random DNA mutations a tumor happened to accumulate. If it didn't have enough mutations, immunotherapy just wouldn't work.
What now?
Now, we know we can pharmaceutically force a tumor to reveal a massive hidden library of non-mutational neoantigens. These hidden flags, from frame shifts, retained introns, and pseudogenes, they can be exposed simply by temporarily cutting the brakes on the NMD pathway.
That's incredible.
It is. The potential value of this for creating highly personalized cancer vaccines and engineered cell therapies is just staggering.
But, as with all breakthroughs we cover, we have to look critically at the risks. Biology is rarely a free lunch.
That raises a very important point and a limitation that's clearly noted in the text. The NMD pathway isn't just a cancer thing, it's a normal, essential biological process in healthy cells.
Right.
We know from human genetics that congenital NMD defects, so where people are born with a broken shredder, cause severe autoimmune diseases and neurodevelopmental issues.
Because if the shredder is permanently broken everywhere, healthy cells might accidentally display weird, harmless typos and get attacked by their own immune system.
Precisely. Therefore, the key to this therapy, eventually making it to the clinic, will be acute transient inhibition. You don't permanently break the shredder, you just inhibit it briefly, just long enough to flood the tumor's display windows and wake up those exhausted memory T cells. And then you let the pathway return to normal to avoid chronic systemic inflammation. It requires precise timing.
It's a hit-and-run tactic to wake up the immune system. We've spent decades trying to build drugs that act as smart bombs to drop on cancer cells. But what if the future of oncology isn't just about dropping bombs? What if it's about breaking the cancer cell's mirror, forcing it to look at its own ugly mutated reflection, and letting your own immune system do the rest? Next time you read about a low-mutation cancer or a tumor that resists treatment, remember the cellular shredder and ask yourself, what is that tumor hiding?